All right. Thank you for your patience. This is probably the least organized talk you'll have at the school because I'm a little bit less experienced at all of the other matters you'll see. And also because we're trying to do a practical demo away from the lab, which is where I'm used to being with all of my nice equipment and piece acquired, and also usually prepare the DNA and I try and use it fresh. Well, and to be honest, how I usually perform the sequencing is, Kevin, can you please do the sequencing? And the magic happens. So we will try to launch the demo first, which wouldn't make too much sense to you. And then in function of how much time there is left, I will try to do some explanation. But more importantly, you'll hear from Dominique Levenier just after me, who will try to then correct my super fast presentation into something which will make pedagogical sense. And you'll hear a lot tomorrow from Pascal Barbry, who is actually the expert in sequencing, unlike me, and I'm very sorry to spoil some of the things. Just a one slide idea of nanopore sequencing. We'll be using a device designed by Oxford Nanopore, and like I just stole the slides from them, is that by some way or form you have DNA that gets attached to this nanopore, as you might have guessed from the name, that is guided through the spore, and it induces some fluctuations in electrical current, which you can sort of see depicted there. That is recorded and then through some computer magic is converted into the ATGC sequences as biologists were used to look at. And you can see the thing being schematically depicted here. So without further ado, let's try and look at what we have here. We have a little cheap device called the flungle, which is this thing over here, and Michela has an older one we've already used that we can pass around for people to look at.
If you can try to make it go to the back and then...
every so often I have to restart the video like this because I don't know why it doesn't work long term. I think it's not right. Oh, there we go. And the magic is that that thing is very portable, reasonably robust, but not very, and economical. So we can bring it to Cargèse, and also when we're prototyping experiments, we can use it. So let me just super quickly check whether it's actually in good shape. Yes, it has some pores for us to sequence, which means that I can safely prepare it and then launch. So when it is supplied to us, it comes with this green storage solution to keep it nice and fresh. It contains some redox buffers and things like that. So we'll quickly flush that out with a solution which is called the flush solution. And at the same time introduce little tether proteins which try to grab DNA and guide it to the pores so that then it goes to the pore and give us some data. The worst enemy of the proteins is drying out without air. So the whole device is sort of enclosed in the microfluidics, and then it's important to not... So I only tested this idea of QuickTime. Maybe if you press, or if I press record, it will be better. Let me actually do the recording, because maybe if I record, it will not want to stop. Oh no. So obviously when you practice, things work, and then they don't when it's the real thing. So let's wash our little device and make sure we don't introduce the air. And unlike the conventional pipetting some of you have seen before, we're going to inject the liquid slowly, and maybe you can see on the camera that there is some liquid going around the contour of the device. So this is the space air, the empty space, which is sort of built in for us to be able to flush some liquids through the device without the whole thing exploding. So let's speed up. And leave a tiny droplet behind so that we don't introduce the air, or not too much of the air. And now let's add the DNA, which I've prepared before and added the adapters and did all of that stuff. And then in the last minute, I mixed it with what they call the sequencing buffer, which I think contains some energy solution for the proteins to guide DNA through the pores at a nice and well-mannered pace. And also you might see that there is some white stuff in the tube. So these are library beads, and the nanopore guys do not tell us exactly what's inside, but they are meant to fall down in the flow cell and then try to attract the DNA closer to the pores, increasing the chance of a DNA coming to the pore and then going through it. So let's put that in before the beads sediment again. And hopefully we can see some white stuff going into the flow cell. Every last drop might count, or maybe not. And let's close the device. So with some luck, this will give us the data for the challenge Dominique will present in a bit. And thank you, Kevin. I think we're done with this part. And I totally forgot that I reset this stuff, so you'll also see me launch the thing. Cargèse challenge, let's record this experiment. I don't know, secret message. Let's check the ID. Yep, that's the right one. We're doing the classical ligation sequencing. Let's quickly check that everything is simple here. Maybe let's not make my laptop work too hard. That I misconfigured that, but so this is the part I thought I planned differently, but I remember where I actually want to store the data, because now that we can write here, and I do actually have enough space, although it doesn't look that way. And then we will also keep the raw data in case we want to play around with that. And let's avoid compression. So that's it. And I maybe should have tested this part before, because apparently I do not have access to the directory, or is there another one? So this is strange because I use a sudo to, there we go. So min no, I think doesn't want to play too nice, but it should write, and we will check that in a second because I did give it the rights. So the device is now warming up, and in a little bit we'll see the interface, and we could then see the pores doing the magic. And in the meantime, I'll clean up. Yep, there we go. So this is the schematic representation of the little green cell you have seen hopefully passed around. And I think right now the device is rechecking itself. And the interesting stuff should be happening momentarily. In the meantime, I can close the video. So the secret message will hopefully appear here. And we can write, so we're safe on that side. So we just need to wait a couple of minutes for the pore scan to complete, giving me time to clean up. Any questions as to what's happened so far? Dominique, I'm sorry to leave a little bit of water around.
Maybe I have a question.
Yes.
So why do we need to be at this temperature?
So I think the Oxford Nanopore people are the ones who've done extensive testing, and they show that like for different stages of the run, they use different temperature. I would guess it is something about balancing the stability of the protein, but also favoring diffusion and dehybridizing the DNA. But this is just my super wild guess, and maybe Pascal might know more. No? Sorry, I distracted it. Would you know why Oxford Nanopore likes a specific temperature profile during the sequencing?
Well, isn't it just because it's proteins that needs to operate at this given temperature?
I also assume so.
Yeah.
Are we still doing the pore scan? Or are we? Yes. 5?
Yeah, around 5.
This is not very fast. It's also not encouraging that it doesn't see any pores. Did I kill the device?
You had 73 before.
But maybe I injected air. I've never done it in front of other people, so performance anxiety is a thing. 5 minutes.
Yes.
It's an eternity.
When I start stronger, I go away for like 30 minutes just because otherwise I'm scared.
Okay, I feel you.
Exactly like Vasily at this moment.
I usually clean up, so I didn't notice the time passing by, which is what I try doing, but I guess when you're...
It's 15 minutes.
It doesn't show me the status anywhere. And I don't want to miss the start, but maybe, yeah, so for this ligation sequencing thing I mentioned, what I've done before coming here is I came in on a Saturday and took the message encoded by Dominique and then added some phosphate groups and then A on 3' ends of the DNA, and attached these adapters I mentioned before that guide the DNA to the pore and then make sure it goes through the pore at just the right space. And schematically, and all of this comes from the Nanopore website, and you're welcome to read up more. They have this sensor chip which broadly holds the pores, and then there is the bulk compartment which is pre-filled with the storage fluid to keep the pores stable over time. And what we've done during flushing is replace it with something more DNA and protein friendly, which is used for sequencing. And then I think the current is applied to pull DNA through the individual pores into those little wells. And on a microscopic level, it looks something like this, which I didn't know until recently, and I think it's really impressive, especially given that they did it like 10 years ago. They then cover it with a lipid layer to hold them the pores, and they are grouped, at least on the bigger device, which you see on an image, but I think it's the same principle for everyone, in sort of clusters of 4 to have some redundancy. So now that I've gone through my reserve slides, is it running yet? Still no?
Vasily, you have your hand raised in the
Sorry, yes.
I didn't see. What was the scale of the image?
50 micrometer.
Okay.
And it's pulled from here. I think this page is not super trivial to find because they published it like 10 years ago when they tried to advertise the original devices. Because if you look at the images of the devices themselves, they're like super out of date, but this is the best I could find. This is not good. Oh, thank you. Such a relief. So here you can see the little representation of our grid, and green means you have a pore waiting for DNA, and bright green means it's doing the God's work. Sorry, we're in France, but it's a turn of phrase. Because our DNA is super short, there is no time for the animation to go and show us that we're sequencing, but actually if we look at these guys, by just comparing the pores available to the ones that are doing the sequencing, we can see that actually most pores that are there are doing something. And usually in the beginning of the run, it's not super, it's not perfect. And then the occupancy picks up. I mean, ideally we want 90% or over, but with a DNA which has been around for a couple of days, I will take whatever. What other cool screens do we have? We can see the size of our DNAs, and we have a mix. I prepared a mix of two things for redundancy in case one of them doesn't work. That was a modification to Dominique's design. And then we can see that there is a certain speed with which DNA goes through the pore. Certain Q-scores, but we'll talk about that later. And then I think the interesting stuff is happening here, where we can see the current, the actual electrical current representation. You can hear that I'm a biologist, so I don't know what I'm talking about. I think, why did I lose a big one? Rescaling. So maybe it froze again. But this is what I usually find most exciting. And this would be it from me. I'm not sure why. Yeah, so the occupancy has picked up. We're now at 73%. We should get some nice data. And this was my last try to get the electrical traces, but I hope you all briefly saw them. So that's on me. I will be distributing the data once I have it. Yes, Serge.
question. Is there a graph that shows the number of reads as a function of time?
Yes.
How does that evolve usually? Is it like monotonous, or is it fast at the beginning and slow at the end?
That is a good intuition. I think if we look over here, so we can get the cumulative output, but also because we're trying to base call in real time, it tries to classify the sort of nicer quality reads and worse quality reads. And what I usually see is that you have a certain amount of time where it's linear, it's sort of full capacity. And then for flongles, which are a little bit more stable, after 6 hours, the stuff starts sort of plateauing off and goes almost flat after, I don't know, 8 or 10 hours. This is, I'm guessing, mostly related to the pores themselves, which gradually get destroyed by DNA going through, decaying. The larger devices last up to 3 days, but we also see this thing that for the first day it's quite linear, and then for the other two it tails off, and we get as much data in the first 12 to 24 hours as in the remaining 48.
So it plateaus because some of the pores die, or?
Yes. Mostly. So you could speculate that you run out of energy or you run out of DNA, but what we see is that usually this pore scan, which from time to time checks how the pores are doing, you start seeing less and less pores being available because they are a protein thing, so they're not super stable to begin with. And I think when you're constantly pushing DNA through them, they somehow destabilize. Yes.
In your graph, in the previous graph that you showed, are the failed reads the ones that have Q-score less than 8? Or are they ones that
Precisely, yes. So it's an arbitrarily defined thing, which I just haven't touched as a preset. These are the Q-scores we get from this fast base calling done on the machine. So maybe we're expecting slightly better Q-scores when I'll re-base call the data with a more computationally safe model, model outside. And then also, I mean, it's a little bit of alchemy, but we noticed that if the DNA is not fresh, and if the device being used was not stored perfectly, then we get lower quality, because presumably there is another mode of pore degradation where it doesn't just fail and give up, but rather for some reason the signal it provides is a little bit worse.
So, what accuracy does Q-score of 8 represent?
Anyone wants to save me on that one? I remember there is an...
It's either 20 log Q or 10 log Q, something like that. There's a mapping between the Q score and the average error rate you can expect per base. And that's a logarithmic function of the Q score, but I can double check this fast, but if anyone knows more information about this. They're correlated, like the higher the Q score, the more accurate you expect the read to be. But this function is a logarithmic function. I can give you more details.
And it's a derivative from the model, right? The base call. It's
Almost.
not fixed.
It's not universal.
Yeah, exactly. It's not universal. So it depends on the base call or the model that you're using. The score is also specific for each.
Yeah, I'm not an expert on this, but this is what I know.
So we can now... So the cool thing about these Q-scores that it provides is that nanopore kind of tells you, okay, that base I'm giving you, I'm confident in. That one I had problems mechanistically reading. And for example, we see that usually the Q-scores at the edges of the DNA, when it just starts going through the pore and when it pops out, are worse. And then we can say, okay, we're just going to ignore that part. Or if in a particular read there is a region which has a bad Q-score, but then we read that DNA multiple times, we ignore the one which has poor information about that area and rather focus on ourselves. So this is the raw electrical signal we get. And then by the magic of machine learning and GPU usage, we can convert this into the biological signal. Again, there are probably people who know much more how it works, but I just take a package provided by the commercial supplier and press go. Dominique, would you like to now make sense of why we're doing all of this?
It's up to you.
Yes, I mean, the thing is launched, so it will just keep going like this for at least 6 hours. Maybe if I can move around with my laptop, I'll keep it overnight.
Yeah, in the meantime, so the probability that a base is wrong is 10 to the power minus q over 10. So, and the quality score is minus 10 log of q. So this means that for a q score of 10, then this is 10 to the power minus 1, which is 0.1. So for a q score of 10, the probability that a base is wrong is 10%. And the more you
Vous
increase
avez
with the q score,
l'adaptateur
you
avec
expect
le
more
cliqueur
accurate
aussi
bases.
qui est là.
So this is the soft information that comes from here, like the Q score of each read. I think we have Q scores per position also, right?
Yes.
So
That's the average, right?
that's the average over the entire lead.
Exactly, yeah.
Yeah, we have Q scores per position in a fast Q file. So for every base, we have some reliability score, which is the probability that it's correct or the probability it's wrong based on this. Yes.
The probability is calculated from the model, what it expects the electric current to be.
Model.
Yeah.
Okay.
Model.
Yeah.
I thought that it was not a single base that is read, but the K-man. So the probability is for, is it for only one?
The probabilities for the base it tried to infer from the K-mer.
Okay.
So the raw data is acquired by K-mers, but then it says, okay, that guy was in the middle.
Okay. And there is one read per K, or maybe we will know this. So you get a K-mer, then everything is read, and then you shift by K or you shift by one?
You shift by one.
Okay.
So assume the K-mer contains 5 bases. I'm not sure what they use right now. Then like 5 frames give you one base, but then they are all contributing to. Je vais juste assurer qu'il ne va pas se mettre en veille, désolé. So Dominique will now explain to us why all of this is being done.
So what I'm going to do now is to explain the challenge and what you will have to do next Thursday. So maybe the best thing is to introduce the pipeline of the coding, decoding schemes, and to focus of the work you would have to do. So at the beginning, you have a mystery messages, which is a message encoding into a text file. And then there is a coder, which is a Python script. I will give you some details about it in a few minutes. And then what you get after encoding this message is a list of oligonucleotides encoded in a FASTA file. Okay. And these oligos have been synthesized, I think, by IDT. And on the experiment we have just done before was the sequencing, which has been done using the OS4 nanopore technology. And then with base calling, the base calling is the operation to transform the electrical signal into DNA sequencing. Then, if everything goes well, after this experiment, we will have a FastQ file, which is what have been sequenced and translated to electrical signal to DNA sequences. Then this file will be the input of the challenge. Okay, and what you have to do from this FastQ file is to process the data to end up with a text file, which is the sequence which has been encoded into the oligos. And then from this text file, which represents the sequence, you can decode it to recover the mystery message. So let's just focus first on the coder. I want to explain how this message has been encoded in order for you to be able to decode it properly. So, the coder. So, as I said before, we start with a text file. And we will encode this text file through this code, this coder in Python. And what we need is the text and also some primers which has been used for amplifying the oligos before. And so, as I said, this coder provides I made a mistake. Is a FASTA file here. So what you need to know is how this message has been encoded. So let's have a look to the oligonucleotide structures, which is as follows. So oops. So the message has been cut, has been split into a bunch of 16 characters. These characters are encoded into ASCII. An oligo, so you have the message. We split the message into 16 characters. And each bunch of 16 characters are encoded as follows. So one character is encoded into 8 bytes, following the ASCII code. And when you got the ASCII code, I made a very simple transcoding into the binary encoding to quaternary encoding. So the 0, 0 is encoded as a A, 0, 0, 1 as a C, and so on. So for example, if you want to encode the S characters, the hexadecimal encoding is 5, 3. The binary encoding, the corresponding binary encoding is what you see here, 0, 1, 0, 1, 0, 0, 1, 1. You can directly transcode into ACGT. So this is the way the oligonucleotide, the payload has been transcoded. Surrounding the payload, there is an index just because the message has been split into several parts. So the index is the location of the 16 characters. So 0, for example, will be the first 16 characters, and so on. And at the end, I added a checksum here, which is a number saying that if the payload plus the index is correct. So this is just to check that if your decoding will be okay. You will have a functions taking these 64 nucleotides plus 4 nucleotides, and then it will produce 4 nucleotides saying that it's okay or not. So this is the oligonucleotide structures you need to know to decode the message. So the decoder takes as input the sequence you will have to decode. It takes as input so the payload plus the index and the checksum. And then if you put these sequences into as input as the decoders, then you will display the message or not. It depends if your decoding is okay or not. So right now you will be able to test these coding, decoding steps.
Can you click OK, Dominique, for this?
Okay.
Thank you. And close this.
doesn't want to. Oops. Okay. Okay. So, if you want to test the pipeline without the message, you can use the coder on the decoders. You can input everything you want in the input of the coders, and then just replace all the chemical parts by suppressing, for example, the commands of the FASTA queue or the FASTA files, and then removing the primers. So you get the real sequence you have to decode, and then you can fill this into the input of the decoder, and you will get the message. It's just if you want to see if it works correctly. Okay?
I have a quick question. Do we know the size of the message?
No.
Okay.
No. It's not a long message. I don't want also to say how many oligos you have. This is something you will have to discover. Yes?
Yeah, the checksum only does error detection, so...
Yes, only error,
correction.
no corrections.
Okay.
But you will see that the coverage will be enough for correcting errors. Just because the message is quite small, so you will have enough information normally to recover the message. Yes?
I have two questions. So when you showed this oligo structure, you showed prefix and suffix.
Yes.
So what are used for?
Yes.
I missed that.
Yes. I don't know exactly, but Vasily can answer the question if he's.
So if we're referring to the same thing, those are just full primers. They were also designed by Dominique, and they just primer landing sites. And then you might remember from this histogram of peak distributions, I said that there is a mix of two things. And the longer reads are coming from a situation where I used longer primers just to generate longer DNA.
So just to increase the quality, you mean, right? Just to increase the quality?
Just to increase the quality, but also because, as you might see, that structure is quite short, and I was afraid of losing it in the process of preparing the library for sequencing, which didn't happen when I did a practice run, but from the combination of the techniques, we can see that the shorter guys disappeared as I was preparing the DNA.
And another question is, what if the checksum is also wrong? What is an error in the checksum part?
If there is an error in the checksum port, then you will see that the message is wrong. The decoding is wrong. Even, to have the checksum okay, everything must be right. If there is an error in the checksums, then it will be wrong.
Either in the payload plus index or the checksum, the suite would be removed, right?
No, the checksum is for checking this part, the payload plus the index. But if the checksum, you decode the wrong checksums, then you will say that this is the limit of the operation, but you won't be able to know if the message is correct or not.
So there might be a case where the checksum is wrong, but the rest is correct. What about the way around?
Yeah, sure. No, but I'm wondering more if the checksum is wrong, but the payload and index is correct.
It can be correct.
Yeah. Omitted, right? Why is point? Because it's not an agreement.
I guess it's up to you. You can discard it if you want. Probably that's the best thing that can be done.
But I've made a website. Everything is explained how the checksum is computed, so you can bypass this part if you want. Okay. So as I was saying, if you want to check the rule coding decoding without the FASTQ files generating by the experiment we have done just before, you can do that. And just to finish, I have a website. Everything is explained. How the oligos is determined. Maybe I can move to it just to show you. I lost it. So, up. I just want to connect to. Okay. Oh, non, il est là. Okay. So you can connect to this website, and everything is explained, how to use the coders, the decoders, and what you can do is to load the Python scripts and everything, and you can try everything you want. And you also have the data, I mean the primer. You need to encode the message. And there are some examples you can use. So what you need is to, of course, internet access, Python on your laptop. And the winners of the challenge will be the guys who will first decode the message. If there are some ex aequo, then we think that we can use some other criteria, such as the method using as less as possible reads to decode the message. And if again there is some ex aequo, then we can test the speed of the method. I mean, okay, so that's all for me. If you have some question, so you can connect to this website. Just take the URL, okay. And yes, the last thing you will need to have is this file here, the mystery.fastq files, which will be the results of the experiment we have just done before. Okay. So right now we do not have this file, but on Thursday morning I will make a link to these files, so you will be able to test your code. On Thursday morning, Thursday morning, yes, I will give you a talk on the way to decode all this stuff. I mean, the different methods you can use to make this bioinformatics decoding parts. Any question?
Will you explain the difference, like why using different graph approaches rather than clustering? Or on Thursday?
Yes. I think there are different methods to decode these things. So the basic method is first to make a clustering of the reads, just to make all the copies of the same oligo into a cluster, and then to try to find out a consensus on these clusterings. This is one possibility. I will explain on Thursday morning some other possibilities to decode these kind of messages.
So we have designed the program of the school so that you'll learn some stuff in the coming days, and then you can put it to practice with a challenge. But we thought we would present the challenge at the beginning so that you have this at the back of your mind as you're listening to different presentations, which will be relevant to the challenge in the end. And also we need some time to make sure we generate the data and then pre-process it for you, so it also helps us.
But if you want to test right now some ideas you have, you can generate some kind of FastQ files with some errors in the reads, and just you can test your method if you want. It's a possibility.
And the primer sequences, they will not be provided as well?
Yes, they will be provided.
Oh, they will?
Yeah.
I think the primer.fasta, right?
Those
okay.
they are here. So, and also I think the suffix and the prefix is something you also you will know it. Okay. So you can download the code. There are some library and some also some useful functions you can use.
Yeah, I have a question regarding the checksum. So here it is 4 nucleotides, so it means that it is some linear combinations of the...
Actually, it is, I take the payload, the index, so every 4 nucleotides, I move it to 8-bit values. I add all the values all together, and the checksum is a modulo 256. Okay, so it's a very easy to compute, you know. But as I provide the code, you can have a look to the code how this is computed. But I think it is explained here. No. Any other questions? Yes, Sarah?
Do you have the number of reads per oligo, or it's inferred by the clustering?
The number of oligos?
Of reads per oligo.
I don't know yet because the experiment is ongoing, but an advice I can give you is that the number of reads you will get, the coverage will be very high. You don't need all that coverage. And my advice is just to, for example, to test with 1,000 reads, 2,000, 3,000, but no more. Maybe we will get 100,000 reads. It's too high, you know.
Sorry, I didn't know how difficult it is to decode, so we ordered a small message, and I'm used to producing a lot of reads.
Yeah. So the advice is to use a small number of reads.
But the coverage of the oligos with the proportion might vary a little bit because they are all produced together in this oligo pool manufacturing, and usually there might be up to 10-fold difference between them. But as Dominique said, in total you'll have so much that it's quite easy to find everyone.
Okay, so good luck.